given me this opportunity. I am a basic scientist, and therefore I would like to understand some pathological problems in Parkinson's disease. And since we are working with different organizations of US government, US Air Force, Swedish Strategic Research and Development, I am from Uppsala University, so I have to say that the views that I am telling here, that is solely to me and not any other organization where I work. This is the classical picture of Parkinsonism, and as you can see that the severe reduction done by MPTP model, we have also followed this MPTP and methamphetamine. Due to lack of time, I am showing only results on MPTP model in mice. What we believe, as I am very happy to hear that the whole brain could be affected, and we believe that blood-brain barrier is the basic cause of all diseases. So when this is affected, we can have neuronal and glial changes in many areas, not only thalamus, but also cerebral cortex as well. I am basically a pathologist trained under Yngve Olsen, and therefore I see this point from a pathological point of view. And we believe that in all conditions of the neurological diseases, this blood central nervous system barrier is disrupted, leading to many changes and causing cell death. And that's why we are proposing that the blood-brain barrier could be a key for neurological disorders. Since we need to not only to treat pathological, but we also have to have neuroregeneration, there are several endogenous factors that can influence neuroprotective elements. And these neurotrophic factors and growth hormones are quite interesting targets. And as well, you can see that nitric oxide plays an important role. In this model, I will show you some results using nitric oxide synthase. Because this is increased in animal models of Parkinson's disease. And in our another model of spinal cord injury, there is a crosstalk when we applied neurotrophic factors, BDNF and IGF, the downregulation of nitric oxide synthase occurs, leading to neuroprotection. So we use cerebrolysin because this is a balanced mixture of different kinds of neurotrophic factors and active key peptides. And we have some previous experience in different models of brain injury. It is quite neuroprotective. So what we have done here, that we use MPTP and we have given cerebrolysin chronically. This is not nanowired because there is another model of delivery we are using, and in this case we have only examined different isoforms of nitric oxide synthase in our animal model. Just in previous example, I can tell you here that the Th positive cells were significantly improved when we have given cerebrolysin in substantia nigra and striatum as compared to the MPTP toxicity model alone. So this drug is working, and the same thing is also working, helping in biochemical analysis. Due to lack of time, I'm not showing all these things. The point is that we can see activation of glial cells and loss of neurons here in the cortex. We have seen here in this case. And when we have given cerebrolysin, we can see that the activation of astrocytes were much less evident, and the neural structures were much normal. Of course, the nanowired cerebrolysin was much more potent than the cerebrolysin alone. So then we used cerebrolysin and studied nitric oxide synthase, and here you can see that untreated MPTP cases, this neuronal nitric oxide synthase and immunological NOS and also eNOS were increased, that were considerably reduced in almost similar areas by cerebrolysin treatment chronically. On the basis of these observations, I can only say that probably nitric oxide synthase plays an important role in the toxicity caused by MPTP in Parkinson's disease model, and probably chronic cerebrolysin treatment may have some therapeutic value that we need to understand further. These are our sponsors and supporters, and I thank you for your kind attention.
Questions to Dr. Sharma? I'm a clinician, so as a clinician, we have this elusive goal of trying to find neuroprotective agents. So is this particular substance used in some other models? You briefly mentioned spinal cord lesion.
Yes.
Mm-hmm.
We have measured, and also this cerebrolysin is under clinical trial in different models with clinical cases of Alzheimer's disease and stroke and traumatic brain injuries. So it is worthwhile to try in an animal model of Parkinson's disease. We have tested in also MPTP and methamphetamine, and in other models of brain injury or spinal cord injury, at least in animal models.
So thank you very much. Then we move on to the last presentation of this first half of the oral poster session. The paper will be given by Dr. Albani from Italy. The title of the paper is Sleep and Executive Functions, Virtual and Neuropsychological Study in Parkinson's Disease and Sleep Apnea Syndrome. Dr. Albani.
Good morning, everybody. We know that sleep may promote memory consolidation and efficient executive functioning. On the other side, we know that sleep deprivation may affect hippocampus